Okay, so welcome to this video. In this video, what we're going to discuss is ion channels and their involvement in cancer. Okay, so let me outline the structure then for this video. What we're going to start off with is a basic overview of the carcinogenesis process as a microevolutionary process where gradually the clone of cancer cells evolves. Okay, then what we're going to do is discuss ion channels, okay, and particularly what we'll focus on is the different types of potassium channels, because we're going to see many examples of where potassium channels seem to be involved in cancer, okay, but we will look at the structures of other ion channels as well, such as voltage-gated sodium channels, voltage-gated calcium channels, and also the trip channels. Okay, then what we'll do is we'll discuss some broad things that could encompass all ion channels and their involvement in cancer. Okay, so we'll discuss the fact that the electrical potential difference across the cell membrane of a cancer cell is generally quite depolarized, okay, more depolarized than a normal cell membrane would be, and this probably results from an aberrant expression of ion channels, okay, and it seems to have very important roles in the cancerous phenotype. Then we'll also discuss about the oscillating electrical potential difference that you find across the cell membrane as the cell cycle progresses, so we'll have a look at that as well. So those are general points that could involve all of the different ion channels that we're looking at. And then after that, what we'll do is we'll go on to some specific examples of ion channels that have been identified as seeming to have very important roles in cancerous phenotypes. Okay, so we'll start with EAG1, EAG1, which is found overexpressed in 75% of cancers. Okay, so that's the one we'll begin with, and then we'll go on to EAG2, which seems also to have very important properties, and then we'll look at some other examples as well. Okay, right, so let's start off then with the carcinogenesis pathway. Okay, so basically the idea, well, our leading theory for how cancer develops is that you start off with a normal cell, and this normal cell gets a mutation in it or some heritable change that can then be passed on to daughter cells that allows it to over-divide, that allows it to outcompete the other cells in that tissue. Okay, so let's draw this here. So you start off with your normal cell here, okay, which is obeying all of the rules of normal tissue homeostasis at the moment. Okay, so this is our normal cell here. And now what's going to happen is it's going to get some mutation, or more strictly speaking, some heritable change, because it can be epigenetic changes and that are heritable, okay, that can also drive this. But to keep this simple, we'll just say it's most likely a genetic change. But there is still a lot of controversy about the importance of genetic changes versus epigenetic changes in cancer. Okay, but genetic changes are more simple to understand how those can be inherited than epigenetic changes are. So we'll just keep this simple and think about mutations. Okay, so it's going to get some mutation in one of its genes that is now going to allow it to over-proliferate. Okay, so it's going to get some mutation, and this is called a somatic mutation because it's occurring in a somatic cell. Okay, so contrast that to a germline mutation, which is something that's in all of the cells of your body because it was in the original fertilized egg that made you. Okay, so this is a somatic mutation. It's just something that's spontaneously going to occur in this cell, in this one cell in the peripheral body. Okay, so now the genes in this cell have changed, and this now allows this cell to over-proliferate. Okay, at least this is what we think is the first mutation that occurs in cancer. Most likely it's an over-proliferation one. Okay, so now what's going to happen is this cell is going to divide and divide and divide and divide. Okay, so you're going to get a whole population of cells that are now all identical to this first cell. Okay, and they all have this same mutation, therefore, that allows them to over-proliferate. So every single one of these cells is going to over-proliferate. Now let's just talk about what sort of mutations could cause a cell to overproliferate. So there are two different ideas here. Either you could have a gain-of-function mutation in what's known as a proto-oncogene. So let me just talk about this. So a proto-oncogene is a gene which is involved in making cells divide. Okay, so it's a gene that if you express it or if you have high activity in the protein that it codes for, it will cause the cell to divide. It's a pro-growth protein, basically. It's involved in making the cell divide. Okay, when these get gain-of-function mutations in, that can be very dangerous because it can cause the cell to now over-divide, divide more than it should do for normal tissue homeostasis. And when the proto-oncogene gets a mutation that allows it to have a higher level of function, it's then called an oncogene. So proto-oncogenes convert into oncogenes when you get gain-of-function mutations in them. Okay, the other idea is that rather than having a gain-of-function mutation in a pro-growth gene, you could have a loss-of-function mutation in an anti-growth gene. Now, the anti-growth genes are what are known as tumor suppressor genes. Okay, these are the genes whose protein has a role in preventing the cell from dividing, so tumor suppressor genes. So this first mutation that this somatic cell gets is either going to be a gain-of-function mutation in a proto-oncogene, which therefore converts it into a oncogene, so I'll just put GOF for gain-of-function here, okay, or it's going to be a loss-of-function mutation in a tumor suppressor gene. Okay, so the important mutations here are loss-of-function mutations here. Okay, so one of these sort of mutations is going to cause the cell to now aberrantly over-proliferate. Okay, and it's now going to make this clone, as it's called, of cells that are all identical to it, all genetically identical to it, and therefore are all capable of over-proliferating. Okay, right. Now, if this produces a lump that is big enough to actually see and feel, then we might now call this a benign tumour, or at least the beginnings of a benign tumour. Okay, at the moment it is not cancer, note. Cancer needs to acquire a lot more properties than just over-proliferation. At the moment it's just a lump of cells that are over-proliferating. Another piece of terminology that's quite useful is the concept of neoplasia. Okay, so neoplasia is broader than cancer. Okay, cancer refers to the final portion of the pathway that we're going to look at. I will show you exactly where we actually arrive at cancer. Okay, neoplasia is broader than that. Neoplasia refers to the entire process. Okay, so this, well, actually, I'll tell you what neoplasia strictly means. It means new growth. Neo means new, plasia means growth. Okay, so this is a new growth now. When you've got this original lump of cells, this original clone of cells that have resulted from a single cell getting this somatic mutation that allowed it to overproliferate. This is now a neoplasia. Okay, we would call this an early stage neoplasia, or just an early neoplasia. Okay, so this is in the early stages of neoplasia. Okay, cancer is going to be the later stages of neoplasia, as we'll see later on. Okay, right. So, how does this then progress? Okay, well now what's going to happen is in this clone of cells that you've produced that are all over proliferating, one of those cells is now going to get a further mutation, okay, that is going to allow it to again get a selective advantage over the others. And this is why cancer is often compared to a Darwinian-like evolutionary process. Okay, it's all about basically the cells no longer acting for the good of the multicellular organism, but now acting as though they are individual little organisms that are trying to compete for survival against each other. So normally in a multicellular organism like humans, all of our cells are fantastic at working together and not doing what is best for themselves, but instead doing what is best for the multicellular organism. Cancer is really cells forgetting that, basically, and behaving now more like bacterial cells, where they're all out for themselves, basically. They want themselves to get bigger and bigger and bigger and bigger, and they don't care about the multicellular organism. Okay, right. So here we have this clone of cells that is over-proliferating, and basically getting this mutation in this original cell allowed it to beat the surrounding cells. So all the cells that were good, that were obeying the rules of normal tissue homeostasis, those are now going to be outcompeted by these cells because these cells over-proliferate so much. So basically this cell has got a mutation that has allowed it to outcompete the surrounding cells, basically. And now what's going to happen is within this new population of cells, this new clone of cells, which is further along this evolutionary process than the healthy cells around it, okay, you're now going to get one of these cells getting a further mutation that is going to allow it now to outcompete the others. So let me show this. So I'm going to draw the cells slightly smaller now. Okay, so let's say here are our green cells here. So these are all over proliferating, and now one of them is going to get a mutation that's going to allow it to outcompete all the others even more. Okay, so maybe it's going to be a mutation that makes it divide even more rapidly. So maybe another proto-oncogene is going to get a gain of function mutation in it and convert into an oncogene. Or maybe another tumor suppressor gene is going to lose its function. Okay, and this will make the cell divide even more rapidly. Okay, but it could be other mutations besides just ones that are making you divide more rapidly, and I'll discuss those in a moment. Okay, so now we've got this new population of cells, or this new clone of cells as it's called, which has a selective advantage over the old population of cells, okay, and is therefore going to divide more rapidly. Okay, like so. So here is this new population of cells. Okay, so this is now a new clone of cells that is now further along the Neoplasia process. Okay, and then it continues on. One of these cells in this purple clone of cells can get another mutation that's going to give it an advantage, and it will then divide and divide and divide and produce a whole clone of cells that are identical to that. Okay, so remember, all of these cells have this initial overproliferation mutation, so the instant you get one of them getting a mutation, it's going to make a whole population of cells that all have that mutation, okay, because it's already got the overproliferation mutation. Okay, so the idea then with cancer is it's this gradual evolutionary process whereby cells, you gradually build clones with progressively more and more mutations in them, okay, and this makes their phenotypes worse and worse, basically. They become badder and badder cells, basically. They get more and more close to having the cancerous phenotype. So let me tell you about some of the mutations that are going to build up in these cells. So the key properties that you're going to eventually build up are you're going to build up self-sufficiency of growth signals. Okay, so basically normal cells that are obeying normal tissue homeostasis rules, they need growth signals to actually make them divide. They need growth factors and all of those sort of things. Basically, these cells, eventually when they do get to cancerous, they're going to have complete self-sufficiency of growth signals. They don't need exogenously applied growth signals in order to divide. They are completely self-sufficient in producing those. Okay, so self-sufficiency of growth signals, I should say there. Okay, resistance to growth inhibitory signals. So this is all about the fact that they are no longer listening to the tumor suppressor genes. So resistance to growth inhibitory signals. So this one, if you like, was all about having enough oncogenes, basically. This one was all about having enough loss of function in tumor suppressor genes. Resistance to growth inhibitory signals. Okay, so that's another phenotype that's going to build up in these cells as you go along this process. Okay, in addition, resistance to apoptosis. So apoptosis is an extremely important part of tissue homeostasis. Okay, in normal tissues that are obeying normal tissue homeostasis, you need to have the same amount of division and apoptosis. Okay, so if you're going to keep a tissue the same size and you've continuously got cells dividing, you need other cells to be dying basically, and cell death, coordinated cell death, is by a process known as apoptosis. Cancer cells stop obeying these rules. Okay, they become resistant to apoptosis and they're not going to commit suicide just because they're overpopulated basically. Generally what happens in the normal tissue is when the cells become overcrowded, some of the cells start committing apoptosis. Not in cancer. The cancer cells become completely overpopulated and don't start committing apoptosis. Okay, so that's one of the key characteristics of these cancerous cells. Okay, another thing that I want to mention is they also generally show a phenomenon known as genetic instability. Okay, now what does this mean? This means that they have a tendency to actually acquire mutations. The rate at which they acquire mutations is much worse than a normal cell. Okay, so potentially there is problems with DNA repair mechanisms, okay, that allow temporary damage to the DNA to actually not be repaired and therefore stand as a mutation basically. Okay, so at some point along this progression, this evolution of clones, what will have happened is one cell in the current clone will have got a mutation that then meant that it became genetically unstable. Okay, and that maybe was a loss of function of some DNA repair pathway maybe. Okay, and then that clone of cells that it produces, and of course the clone of cells will be produced because it's already got the overproliferation phenotype. So if I just show this on this pathway, well on this picture here, let's say one of these purple cells here, which I'm now going to colour in orange, managed to get a mutation in one of its DNA repair pathways. Okay, that now means that it's not going to repair its DNA when there's damage to the DNA, and therefore that damage is going to remain, and therefore that's going to potentially lead to mutations that would have previously been fixed remaining there. Okay, now what will happen is because it's already got the overproliferation phenotype, it will produce a population of cells that all have this loss of function of this DNA repair pathway. And why is this advantageous for the progression of cancer? Well, now in these cells the progression process will be sped up hugely, okay, because mutations are a slow process. It takes a long time to acquire mutations in these previous clones because the cells are so good at repairing their DNA. Now, in this orange clone of cells, it's going to go much more rapidly, basically. You know, all of these cells will be getting mutations all the time. Some of those mutations will, of course, kill the cells. Maybe if they get a mutation that's saying complex 4, a protein of complex 4, that will kill them, presumably. But some of them then will get mutations that then allow them to go further along in this progression towards the cancerous phenotype. So having genetic instability is believed to hugely accelerate the process after that. Okay, right. So those are the main ones. There are some other characteristics that people talk about with regards to developing carcinogenesis. Okay, so for instance, some people think that some cancer cells will get mutations that help them in the angiogenesis pathway. Okay, so normally what happens is when a portion of tissue is ischemic, isn't getting enough blood supply, isn't getting enough delivery of oxygen, what will happen is it will set up angiogenesis. It will set up the growth of new blood vessels into that area, and that will help to deliver more oxygen to that area. Okay, now if you produce a massive great tumour like this, and it's still a benign tumour as of yet, we haven't talked about how it becomes cancer yet, okay, of course, this is going to become ischemic in the centre of the tumour. Okay, so they will actually start setting up the signalling pathways to get angiogenesis occurring into this tumour mass. However, some people believe, and in fact we will see an example potentially of this later on, that although angiogenesis, you will get it even if you don't have any mutations that promote it, it would be advantageous for the cancer cells to get a mutation that promotes angiogenesis. Even though they don't need it, it is a normal physiological pathway that the cancer cells can use, and they will get angiogenesis. Suppose if they did have a mutation, however, that increased how much they promote angiogenesis, then you'd get more angiogenesis occurring, and that would deliver more oxygen to the tissue. Okay? So that would actually provide the tumour with a selective advantage. So potentially, at some point in this progression, they're going to get mutations that promote angiogenesis signalling, okay, and therefore that's going to help with the tumour survival, because more angiogenesis means more oxygen delivery to the tumour. Okay, even though it's not absolutely necessary, you don't need it, but it might actually promote the tumour if you do get it. Okay, right. So I'll add that one there. Okay, right, so this is this gradual evolutionary process. You're gradually making these new clones of cells that have more and more of these dangerous phenotypes, okay? Now, at the moment, all of this is still counted as a benign tumour, okay? Yes, we're progressing through the neoplastic stages here, but it's still benign at the moment, okay? And the reason it's still benign is at the moment these cells are still not motile, okay? At the moment these cells are static. They are a solid mass tumour, and they're static where they are, okay? What makes this dangerous is when suddenly they get mutations that then allow them to become motile. So usually cells in tissues are not motile, they're not moving around. Usually they have their own little position and they don't move from that position relative to the body, okay? However, what happens in the later stages of neoplasia is that in your final clone in this progression process, what's now going to happen is another mutation is going to occur that allows the cells to become motile. So the final transformation that turns this benign tumour into a malignant tumour, i.e. cancer. Okay, so what we're about to look at is how this becomes malignant. Okay, malignant neoplasia, or cancer basically. Okay, or you could call it a malignant tumour. So the final transformation that occurs is that within one of these cells of the final clone before you become cancerous, you're going to now get a mutation that allows the cell to become motile. Okay, so a mutation that promotes motility. Okay, so the cell is going to now gain the phenotype of motility. So let me just discuss motility a little bit more. Okay, well, most of the cells from which cancer develop, generally they are epithelial cells. Okay, so most cancers develop from epithelial cells. So for instance, the cells that line the gastrointestinal tract, those are all epithelial cells. The cells of the skin, those are all epithelial cells. The cells that line the lungs, those are all epithelial cells. The cells that line the urogenital tract, those are all epithelial cells. Okay, usually cancer develops from these sort of epithelial cells. In addition, we can talk about the cells that line the liver and the pancreas. Those are all epithelial cells as well. Okay, so usually most forms of cancer develop from epithelial cells. It's much, much rarer for them to develop from non-epithelial cells. So for instance, muscle cells, they're very, very rare. You hardly ever hear about skeletal muscle cancer, or smooth muscle cancer, or heart cancer. Okay, those are much, much rarer. So, most cancers develop from epithelial cells. Now, epithelial cells are not usually motile. However, they can get phenotypes which allow them to become motile. And basically what seems to happen is the cancer cells turn on a behaviour that was used in embryology. Okay, so in embryological development, there is a process by which epithelial cells can move around. Okay, and I just want to tell you about this process. So basically, if we have an epithelial cell, let's have an epithelial cell here. Okay, so if this epithelial cell wants to move around, what needs to happen is it needs to change from being an epithelial cell to being a mesenchymal cell. Okay, at least this is how it works in embryology. What happens is you have your epithelial cell here, and I'll just label this up. So this is the epithelial cell. And basically what happens is the epithelial cell undergoes a transition from being an epithelial cell to being a mesenchymal cell. So I'll now show my mesenchymal cell. I'll have to draw it in a different colour so that they don't look similar. So here is my mesenchymal cell here, okay, in red, and then I'll have my epithelial cell here in green. Okay, and basically the epithelial cell can undergo a transition to being mesenchymal-like. Okay, so this is now a mesenchymal cell. Okay, and this transition from being an epithelial cell to being a mesenchymal cell is what's known as EMT. Okay, so this stands for epithelial mesenchymal transition. Okay, so the E is for epithelial, the M is for mesenchymal, and the T is for transition. Okay, so in embryology, if an epithelial cell wants to move around, it firstly needs to undergo epithelial mesenchymal transition, EMT, become a mesenchymal cell, and then this mesenchymal cell is motile and can move around. And then when the mesenchymal cell gets to where it finally needs to go, what can then happen is it can transform back to being an epithelial cell. Okay, so we'll go back to being an epithelial cell here, which I'll colour in green, and this is a process known as mesenchymal epithelial transition, or MET. Okay, so basically this is a process that's used all the time in embryology. Okay, so epithelial cells that need to move around in the embryo, they first transition into mesenchymal like cells, okay, and then when those cells have got to the new destination, they then transition back into the epithelial phenotype. Okay, right, so basically this system seems to be reactivated in cancer cells. It's not activated in normal healthy adults, okay, but in cancer cells they seem to reactivate this process. So what generally happens is you get some mutation that allows these cells to undergo epithelial mesenchymal transition, and these mesenchymal cells are now motile. Okay, and what this means the tumour can now do is it can invade the surrounding tissue. Okay, so previously benign tumours, they were a lump that was separate from the healthy tissue. Okay, they were potentially compressing the healthy tissue, but they weren't invading into it. Okay, because they weren't able to actually move. So what now starts to happen basically is that this tumour, let's say we'll have a new clone of cells here, I'll just produce it. So let's say this was our final clone in this progression, and I should say there are thought to be more clones in between starting from the healthy cell and getting to the final cancerous cell than how many I've shown here, which is 1, 2, 3, and then 4. Okay, it's generally thought that there's about 7 or 8 intermediate clones between going from being a normal cell to being a finally cancerous cell. Okay, right, so here in red then, these are our final cancer cells. These are the ones that have undergone the mutation that now means that they can be motile. And in the case of epithelial cells, it seems to be that they can activate this epithelial mesenchymal transition. Okay, so what's now going to start happening is some of these cells will start undergoing this process. They'll start undergoing epithelial mesenchymal transition. Okay, they'll become mesenchymal cells. They'll then move into the surrounding tissue. Okay, they'll then go through mesenchymal epithelial transition, returning back to the epithelial cell. Okay, but now they're in the surrounding tissue, and they'll then start dividing. Okay, and this means they will set up another little mass of tumour that's further away from the primary tumour site in the surrounding tissue. Okay, so what will now start to happen then is the tumour will start to invade into the surrounding tissue basically. Okay, now that's invasiveness. Okay, that's not like that. That's invasion. There's another thing that occurs once these cells have started to acquire this motility property, which is a phenomenon known as metastasis. Okay, so what can happen is the cells can undergo the epithelial mesenchymal transition, they can become motile, and they can invade into blood vessels, which is what this is supposed to show here, and also lymphatic vessels as well as blood vessels. Okay, so here now, let's say this is our blood vessel. Okay, so what will happen is this cell will undergo epithelial mesenchymal transition. It will start moving, it will go into the bloodstream, and then it will start circulating in the bloodstream. And whilst it's circulating in the bloodstream, there's a special name for that. They're known as circulating tumour cells. Okay, and then finally what seems to be able to happen is the circulating tumour cells can leave the bloodstream at other sites. Okay, and then they can move into those other tissues far, far away, and then they'll obviously undergo the mesenchymal epithelial transition. They'll return back to the epithelial cells, and then they can set up another tumour at this far away site, and that's known as a secondary tumour, which is in a totally different tissue from where the original tumour occurred. Okay, so a secondary tumour. And the other term that's often used for this is to call the secondary tumour a metastasis. Okay, and metastasis is also used to describe the actual process of the primary tumour sending out these invasive motile tumour cells that can then move to secondary sites and set up secondary tumours. Okay, right, so that's metastasis, and generally it's metastasis that kills you in cancer. Okay, generally what ends up happening is you get metastatic cancer cells setting up secondary tumours in the brain, and that is what generally kills you. So metastasis in the brain is generally what kills most people with cancer. Okay, so now that's an overview of what then happens in cancer. Okay, what we now want to talk about is the involvement of ion channels in this pathogenesis process. Okay, now the first thing that I should say is that this is a very new area. Okay, there are hopeful signs here that ion channels probably are involved in cancer. Okay, but it is still controversial. Everything that I'm telling you here, well, what I'm going to tell you here, is going to be very well, still controversial, not set in stone yet, and also it's going to be only very little snippets of knowledge. There are not established huge long pathways like there are for tyrosine kinases and things like that yet. Okay, so a lot of what I'm going to tell you is just snippets of knowledge that may in the future have much more known about them, okay, and much more developed pathways, but at the moment this is still a very new area and it's in its early days. Okay, however, there is a lot of interest by pharmaceutical companies in the possibility that ion channels are involved in cancer. Now, let me tell you the reasons. Well, the first reason that we have to suspect that ion channels are involved in cancer is that when you look at cancer cells, the ion channel expression profiles are extremely dysregulated. Okay, so they have extremely aberrant expressions of ion channels that are not right, basically, that are not normal. Okay, now the big then question is this aberrant expression of ion channels a driver for cancer? i.e., does it drive the cancerous phenotype? Is it involved in one of these transitions between clones? Okay, does it give the cells a selective advantage in being able to outcompete their neighbours? Okay, or is it just passive? I.e., is it just a result of all the other things that are going wrong in cancer cells, and it's just a result of that, but it's not really actually driving the cancerous phenotype? Okay, and that's the big question that remains to be answered. Okay, but I'm going to give you examples of where it looks as though these are drivers, and they do play a part in developing the cancerous phenotype. Okay, right. The other thing to say is, why is the pharmaceutical company so interested in this? Well, it's because ion channels are generally on the plasma membrane, okay, which means that they are a easy target for drugs. Okay, it's much more difficult to get drugs inside cells, okay, whereas if your target is on the surface of cells, that's much easier. Moreover, one of the great revolutions that pharmacology is going over to is the use of monoclonal antibodies that are specific for proteins, okay, because that allows us to have incredibly targeted therapies. Okay, now, antibodies do not get inside cells, so they are absolutely only useful for targeting things on the surface of cells. So that's another reason that ion channels are exciting, because again, they're on the surface of cells, and that makes them pharmacologically tractable, particularly with these monoclonal antibodies that are taking over everything. Okay, right, so that's why there's so much interest in ion channels and potentially targeting them as a cancer therapy, because they are so availably tractable for pharmacological means. Right, so what we're now going to do then is we're going to turn our attention to discussing some of the major ion channel types that we're going to need in our later discussions. Okay, so what we're going to start off with is the potassium channels. Okay, and these are the ones where I've got the most examples of where these are involved potentially as drivers in cancer. Okay, so I'm going to tell you firstly then about potassium channels. So potassium channels are a very big family of ion channels that allow potassium to move across the cell membrane. Okay, now, potassium channels are divided up into 4 major families. Okay, so I'll tell you what these 4 major families are. So the first family is the voltage-gated potassium channels, which for short I'll abbreviate to VGKC. So this stands for voltage, that's the V, gated, that's the G, K for potassium, and then channels. Okay, right, now another piece of nomenclature for the voltage-gated potassium channels, which is important when we come to actually name the subunits of the voltage-gated potassium channels, is to write K for potassium and then V, so you subscript it V for voltage-gated. Okay, and we'll be seeing this notation a lot more later on. Okay, right, so that's a massive great family of potassium channels, which we'll study in more detail in just a moment once I've stated all 4 families of potassium channels. Okay, the next family of potassium channels is what's known as the calcium activated potassium channels, and these are potassium channels that are going to actually be activated to open by the presence of intracellular calcium signals. And again, we're going to see how some of these are involved potentially in cancerous phenotypes, okay? Particularly they're involved in potentiating calcium signals, which we'll see an example of later on. And also they're potentially involved in the actual motility that cancerous cells develop, okay? So we'll look later on more in detail at how cells actually move, okay? How, once the epithelial cells have undergone the mesenchymal transition, how do those mesenchymal cells actually move? And calcium-activated potassium channels, particularly the intermediate conductance calcium-activated potassium channels, may play a role there, okay? So we'll discuss those in more detail in just a moment as well. Okay, next family of potassium channels, okay, and we're getting gradually more niche. The next family that I'm going to mention are the inward rectifying potassium channels, KIR. Oh, and I should just say, the calcium-activated potassium channels are usually abbreviated to KCA, like so. Potassium channels that are gated by calcium. Okay, KIR then, which are the inward rectifying potassium channels. Okay, or inward rectifier potassium channels. Okay, and these can also be denoted as the ERK channels. So I for inward, R for rectifier, and K for potassium channels. So either of those nomenclatures we will see. Okay, right, and we'll discuss why they're inward rectifier potassium channels in a moment. Okay, but for now, basically they have a tendency to allow potassium to come in more than they have to allow it to go out, but that isn't because the actual pore prefers to allow potassium ions in. There's a more subtle reason for why they prefer to allow potassium in than out. It's because under conditions when potassium will be moving out, basically the channels are blocked, and then when under conditions when potassium will be moving in, the potassium channels aren't blocked. And we'll look at that in more detail in just a moment when we come on to discussing these channels. Okay, and then finally, the family of potassium channels that no one's ever heard of, but actually are extremely important, is the two-pore domain potassium channels. Okay, K2P. So this is the two-pore domain potassium channels. Now these ones are the leak potassium channels. These are the ones that are always open, basically, and which are responsible for the establishing of the electrical potential difference across the cell membrane, okay, by the Goldman-Hodgkin-Katz constant field equation. Okay, so two-pore domain potassium channels, also called twin-pore domain potassium channels or tandem-pore domain potassium channels. Okay, do not, and repeat, do not call these just two-pore potassium channels. If you call them two-pore potassium channels, that's something else. Two-pore potassium channels are an extremely putative potassium channel that actually seems to have two pores. Okay, these things do not have two pores. The subunits have two-pore domains. Okay, and I'll come on to what that means later on. Okay, so these things do not have two pores, basically. Those are different things that are much more putative. Okay, so these are the four major families of potassium channels that are established and mainstream in molecular biology. Okay, right. So let's now discuss the structure of some of these different potassium channels. So we'll start off with the voltage-gated potassium channels firstly. Okay, and we're going to see loads of examples of voltage-gated potassium channels later on when we go on to the specifics. Okay, right. So voltage-gated potassium channels then, VGKCs, voltage-gated potassium channels. Okay, so these are in the plasma membrane, so I'll use these two parallel lines here to represent the inner leaflet and the outer leaflet of the plasma membrane. So this side up here, this is the extracellular fluid, and this side down here, this is the cytoplasm. Okay, right, and the voltage-gated potassium channels are going to be sitting in the plasma membrane. So let me draw this here. Now they are not just one protein, so this is the first important concept. In fact, none of these potassium channels are going to be just one protein. They're all multi-protein complexes. They're made up of more than one protein stuck together. Okay, now voltage-gated potassium channels, in fact, are tetramers. They're made up of 4 quarters, basically, 4 separate subunits bound together. 1, 2, 3, 4. So they're a tetramer. Now this picture is oversimplified, okay, but it's oversimplified helpfully. Okay, we'll come on to a more complicated picture of voltage-gated potassium channels later on. Okay, for now, the thing that you should take from this picture is that you have these 4 subunits that make up each a quarter of the voltage-gated potassium channel. Okay, now, what I want to do then is pull out one of these subunits of a voltage-gated potassium channel, here in purple, and I want to have a look at its membrane-spanning topology. Okay, so let's have a look at the structure of what a subunit of a voltage-gated potassium channel. Okay, so again, here is the cell membrane, and again we'll have this as the extracellular fluid side, and this is the cytoplasmic side. Okay, now what we want to have a look at is how this single subunit, which is a single polypeptide, how does it span the membrane? What is its membrane spanning topology? Okay, so its membrane spanning topology is like so. You have a first cluster of 4 membrane spanning alpha helices, which I'll draw here, and this is the amino terminus intracellularly here. Then after those 4 membrane spanning alpha helices, you have a large linker there, and then another membrane spanning alpha helix, a p-loop, and then another membrane spanning alpha helix, and then the C-terminus here. Okay, so let me highlight up some of the different portions that you have here. Okay, so firstly you have 6 in total membrane spanning alpha helices. Okay, and I'll colour these 4 in one colour, and these final 2 here in another colour. Now, these are named S1 is the first membrane spanning alpha helix here, then S2 is the second, S3, S4, S5, and S6. So they're named S1 to S6. Okay, right. Now the reason I've coloured these 4 in here, a different colour to these two here is that these are actually separated from these two here, actually in the physical structure of the voltage-gated potassium channel, and I'll draw you a better structure of the voltage-gated potassium channel in just a moment. Okay, so these four membrane-spanning alpha helices here, this cluster is then known as the voltage-sensing domain of the subunit of the voltage-gated potassium channel. So this is the voltage-sensing domain. Okay, then these two membrane-spanning alpha helices here, with the p-loop in between them, this is what's known as the pore-forming domain. So this is the pore-forming domain. Okay, right, and these are actually going to be separated in the structure of the voltage-gated potassium channel. So let me now draw you a more correct but more complicated picture of what a voltage-gated potassium channel looks like. Now to do this, I'm not going to try and draw it on this odd 3D angle like so. Instead, I'm just going to draw it simply looking from the extracellular aspect down on the voltage-gated potassium channel. So I'm just going to draw you the 2D plane, basically, of what it looks like from above, because that's simpler to draw. Okay, so basically my picture was half right. Part of it does look exactly like what I have just shown you here, where you have these four separate portions making up the pore-forming domain portion here. Okay, but then you also have these additional portions around the outside, and these are going to be the voltage sensing portions. Okay, and you might think, well, that's fine, that's not too difficult. So you'll have one pore forming domain here, and then its voltage sensing domain will be here, but there's a subtlety here. Basically, this voltage sensing domain, this pore forming domain, and it's the pore forming domains that make up the central bit, which I apologise that this portion here is coloured in the same colour as that. In reality, it's the pore forming domain here in orange that makes up these portions here. Okay, however, basically it's not as simple as you might think. It isn't the case that this pore forming domain here belongs to the same subunit as this voltage sensing domain here. No. Basically, this one is going to end up paired with this one. This one is going to end up paired with this one. This one is going to end up paired with this one, and this one is going to end up paired with this one. So if I just now show these linkers between the pore forming domains and the voltage sensing domains, it'll look like this. Here's a linker between this voltage sensing domain, which I'll colour in purple here, which is this cluster of four membrane spanning alpha helices here, S1 to S4. This is its S4, S5 linker, as it's called. So this linker on the cytoplasmic side, which is in turquoise here, this is the S4, S5 linker. Okay, and here's the S4, S5 linker linking this voltage sensing domain here to the pore forming domain here. Okay, now if we continue this on, here's another voltage sensing domain, and it will then be linked to this pore forming domain down here. Okay, here is another voltage sensing domain, and it will be linked to this pore forming domain down here. And then finally, just to finish it off, this is a final voltage sensing domain, and its pore forming domain will be this one over here. Okay, right, so that's one little subtlety to this. Right, so basically this is the portion that forms the pore, okay, and specifically this little loop between S5 and S6, this reentrant loop that tries to span the membrane but fails, this is what's known as the P-loop, okay, and that stands for the pore loop, okay, and in fact this actually lines the pore at the extracellular aspect, so it can't line the pore all the way through because it doesn't actually go all the way through the membrane, but it lines the pore at the extracellular aspect, and it creates a very narrow portion that is there to ensure that only potassium ions can move through this channel, and that narrow portion of the pore is what's known as the selectivity filter. Okay, so here are the P-loops surrounding that extracellular aspect of the pore, and this narrow portion is then called the selectivity filter. Okay, The rest of the pore lower down is lined by S6. Okay, so this is the selectivity filter. Right, okay, so that's the structure then of a voltage-gated potassium channel, and before we move on, let me just say that these are the pore-forming domains. The voltage-sensing domains obviously sense voltage, okay, so when the electrical potential difference across the cell membrane becomes depolarized, what happens is these voltage-sensing domains actually all move outwards. They move towards the extracellular fluid, basically, and this causes a conformational change that is transferred to the pore-forming domains by the S4-S5 linker, and that then causes a change in conformation in the pore-forming domains, which then opens the voltage-gated potassium channel. Okay, right. Now, let's just discuss how many different voltage-gated potassium channels there are, because it is an absolutely enormous family. There are in fact 40 different subunits for voltage-gated potassium channels. In fact, it's worse than that. There are 40 different genes for subunits of voltage-gated potassium channels. Okay, and these genes could have multiple splice variants, and I'm sure some of them do. Okay, so basically, what I mean by that is that the simple idealism, which is that there is just one gene which codes for the subunit of a voltage-gated potassium channel, and you just have to make 4 copies of this protein, stick them together, and then you've got your voltage-gated potassium channel. That's not true. In fact, there are 40 different genes which all code for subunits that look like this, which can make up 1/4 of a voltage-gated potassium channel. Okay, now, these 40 different genes are split into 12 different families. Okay, and let me tell you the nomenclature for the 12 different families. So basically, the 12 different families are the KV1 family, the KV2 family, KV3 family, KV4 family, all the way up to KV12 family. So these are not individual channels, these are families of channels. Okay, so let's now have a look at one of these families to get the picture here. So if we look at the KV1 family, which is quite an important family, it's actually got 8 different members. So 8 of these genes are in this KV1 family. Okay, and those 8 members are called KV1.1, KV1.2, KV1.3, all the way up to KV1.8. So the nomenclature is quite nice and simple, basically. You split these 40 genes into 12 different families, KV1 through KV12, and the members of the individual families are then just KV, whatever family it is. So I'll put KV Y for the family, so Y is just a number from 1 to 12, and then you put point X for which family member it actually is, basically. So here we've got family number 1, and then it's the member number after that. Okay, right, so that's the way that the different voltage-gated potassium channel subunits are named. Okay, now, how do they assemble together? Well, that's a total nightmare. Okay, most of the examples that we are going to see are going to be homotetramers, where you assemble 4 identical subunits together to make a voltage-gated potassium channel. However, there are some of these 40 genes that can't actually form homotetramers. We're not actually, I think, going to see a single one of the ones that can't actually form homotetramers and have to form heterotetramers. So I'll just write these big words out. So homotetramer is a tetramer made up of 4 identical voltage-gated potassium channels. A heterotetramer is a tetramer made out of different voltage-gated potassium channel subunits. As I say, we're not going to see any heterotetramers, so you don't need to worry about that. All the examples that we're going to see are homotetramers, but more generally for voltage-gated potassium channels, there are some of these that can't actually form homotetramers and have to form heterotetramers with other voltage-gated potassium channel subunits. Okay, right. So those are the voltage-gated potassium channels. Now, before we just move on to the calcium-activated potassium channels, what I would like to talk about is a related term that you often hear referred to with regards to voltage-gated potassium channels. Okay, and this is the concept of a KDR. So what is the concept of a KDR? Well, this is the concept of a delayed rectifier potassium channel. So the DR is for delayed rectifier potassium channel. So basically, this is one of these potassium channels that is extremely important in the action potential. It's extremely important in the downstroke of the action potential. Okay, so remember, in the action potential, if I just draw a little picture for this, what firstly happens is when you reach threshold potential, you firstly activate voltage-gated sodium channels, which I know we haven't discussed yet, but we will come on to. You firstly activate voltage-gated sodium channels to open when you get up to threshold potential, and they open very, very quickly and allow sodium into the cell. Okay, and this causes that upstroke of the action potential. So if I just draw a little graph here, okay, here is resting membrane potential. You then have your initial depolarization up to threshold potential, and then when you get up to threshold potential, you open the voltage-gated sodium channels very quickly. Okay, and that causes the upstroke of the action potential. Okay, so that's what we're looking at here now. Okay, then what happens is the voltage-gated sodium channels inactivate, okay, and therefore close, so the sodium current that was coming in stops, and then what finally happens is the voltage-gated potassium channels open. Now, these potassium channels, these were actually opened to activate at threshold potential. They were activated to open at the same time as the voltage-gated sodium channels. Okay, so this is our voltage-gated sodium channel here. Okay, so they just take absolutely ages to open, and by the time they actually get round to opening, the voltage-gated sodium channels have opened and have actually inactivated by now. Okay, so these potassium channels, these voltage-gated potassium channels involved in the action potential, they are called delayed rectifier potassium channels because of how delayed that opening is from when they were initially actually activated to open. Okay, so at this point they will then of course open and allow potassium ions to move, and I'm sorry it's not open, but I don't want to have to draw another picture. So it will open and allow potassium channels out, and that will cause the downstroke of the action potential. Okay, so the concept of a delayed rectifier potassium channel then is one that takes a long time to open from actually being activated to open. Okay, now the major delayed rectifier potassium channels, which are expressed in neurons and are responsible for the action potential, are voltage-gated potassium channels in the Kv1 family, so the Kv1.x voltage-gated potassium channel subunits, and also the Kv10 family. And we're actually going to see the members of the Kv10 family later on. Those are the ether-a-go-go voltage-gated potassium channel subunits, and they're actually implicated in the driving of carcinogenesis phenotypes. Okay, right. So that's what is meant by a delayed rectifier potassium channel. It's a concept that comes up all the time when you talk about voltage-gated potassium channels. Okay, right. So we'll call it there for this video, and in the next video what we will do is we'll start by looking at the calcium-activated potassium channels. Okay, then we'll look at the inward rectifier potassium channels and the two-pore domain potassium channels. Then we'll look at voltage-gated sodium channels, voltage-gated calcium channels, a little bit on trip channels, and then we'll start looking at the individual examples of where some of these ion channels may be involved in carcinogenesis.